So once we get from the mouth, um, tongue, the teeth, mastication, chewing, start the initial stages of digesting, get to the pharynx, to the esophagus, and eventually to the stomach. And this is where things really start to get sort of complicated, but also very interesting. All right, so the anatomy of the stomach, the esophagus pierces the diaphragm. So remember the diaphragm where your heart and your lungs sort of sit on your diaphragm. The esophagus pierces the diaphragm, passes through the esophageal hiatus to empty into the J-shaped organ known as the stomach, which leads to your stomach. Primarily in the left upper quadrant, right? Convex side is called the greater curvature. Concave side is called the lesser curvature. Convex, concave. All right. So the stomach has 5 regions that we break it up into, right? So the cardia region, not necessarily, it would be the region closest to the heart, but not necessarily, don't necessarily think of cardio, like cardiovascular, cardia region, where this is where your esophagus, so your esophagus tube, posterior to the trachea, goes down to your crosses, so it goes through the diaphragm, goes into your stomach. All right, this is where the esophagus empties into the stomach. This is where the bolus that you've chewed up food enters into your stomach. Then we have the fundus, dome-shaped top of the stomach, the body, the large portion of the stomach, phyloric antrum, inferior portion of the stomach, and then you have the phylloris, a terminal portion of the stomach, and this is going to lead to your duodenum, which is the first portion. We're gonna talk about the different portions of the small intestines, but the duodenum is the first portion of your small intestines. All right, so these are the parts of the stomach. The pylorus contains the pyloric sphincter. And see, notice another sphincter here. So we have places where you have closing off of, from the pharynx to the esophagus, from the esophagus from the esophagus to the stomach, the gastroesophageal sphincter, and then we have the pyloric sphincter from the stomach to the duodenum, to the small intestine. So things open and then close off so that food does not go in the wrong direction, right? We want everything basically flowing from superior to inferior, right? Now, when you go through the intestines, you kind of go around, so it is a little bit going superiorly again, but for the most part, from one end to the other, right? The interior of the stomach contains these folds, rugae, allow stomach to expand considerably. You ever, when you eat, notice how you feel like full? You not only feel full, but you look bigger sometimes in the stomach, depending on how much you've eaten, especially Thanksgiving. Your stomach has the ability to expand. One of the, and I was just thinking, this just popped into my mind, but one of the ways we try to limit or help somebody lose weight is by reducing the ability of their stomach to expand. The name of it is slipping my mind right now, but if we can reduce the ability of the stomach to expand, if we allow it only to get so big, then only a certain amount of food can get into your stomach and you won't eat as much food. That's the idea of the device that does that, which I can't think of right now. I'm sure somebody watching this is saying it. It's on the tip of my tongue, but I'm not getting. Anyways, it'll come to me. right, so this is the picture of of the stomach, gastroesophageal sphincter, allowing the bolus to enter the stomach from the esophagus. Notice we're below the diaphragm. Esophagus comes through the diaphragm. Fundus, cardia, the main body, pylorus, the end, and another pyloric sphincter at the end. And this is what it looks like in inside a cadaver, cut open version of the stomach. All right, so what is the histology of the stomach? Same four tissue layers as the rest of the alimentary canal. Mucosa, it's your sort of epithelial layer, submucosa, connective tissue layer, your muscularis externa, which is your smooth muscle, and your serosa, which is also primarily connective tissue. Right, the muscularis externa of the alimentary canal usually has two layers of smooth muscle, inner, circular, and outer longitudinal. There's an additional inner layer of smooth muscle in the stomach's body, but this fiber is oriented obliquely. This oblique layer of smooth muscle allows for the stomach to perform churning motion, which helps pummel the food into a liquid called chyme. Right, so we go from bolus to chyme at the stomach as we further churn and break down the food. Mucosa of the stomach is heavily indented to form deep structures, gastric pits. We have columnar cells, goblet cells, which secrete mucus, help protect the lining of the stomach from the acids that it's very acidic in the stomach. You have gastric glands at the base of gastric pits, which the idea here is that these are multiple branch glands that contain both endocrine and secrete hormones into the bloodstream, estrogen cells that secrete an acidic enzyme containing fluid called gastric juice into the lumen of the stomach. And this is that helps decrease the acidity of the stomach. Remember, this has to stay in the stomach. We don't want this acidic enzyme containing fluid known as gastric juice to get into other parts of our body. It is necessary for the breakdown of the food that we have eaten. All right, so this is the structure of the stomach. Notice these gastric glands in these gastric pits. We have the secretion of mucus on the top for protection. This is the mucosa layer of the stomach. We also have the submucosa, the connective tissue, which is going to have our blood vessels. Muscularis, we have 3 layers of muscles, and then serosa, sort of the surface. 4 main types of cells in the gastric glands, each secrete different products. We have mucus neck cells, located near the top, secrete the acidic mucus. Prevents neutralization of acid produced by parietal cells. We have parietal cells which secrete hydrochloric acid, a very strong, very acidic acid. It's responsible for the acidic pH of the gastric juice. This also activates pepsinogen, right? We're going to see that when we talk about a little bit more and go into the digestion. Also destroys many disease-causing agents. Once again, we have another sort of aspect of the immune system available here. If something were to get past the saliva, the immunoglobulins, the lysosomes in your mouth, the malt tissue, the tonsils, down to your esophagus, down to your stomach, it would still have to encounter hydrochloric acid once it gets to your stomach, which kills a lot of things. So that pathogen that has entered your body, when they say don't put your hands in your mouth, there are a lot of ways even if you did put your hands in your mouth, that your body could fight off those pathogens. I'm not telling you to touch things and put your hands in your mouth, but I am telling you that your body has many ways to fight off pathogens. All right, so we have the activating of the pepsinogen. That's going to be very important because that's going to be very important when we talk about protein digestion. Destroys disease-causing organisms, stimulates parietal cell production of chemical intrinsic factor required for the absorption of vitamin B12. That's all your parietal cells. We also have chief cells, another type of cell that is that is at work here. Secretes an inactive precursor enzyme, pepsinogen. When this pepsinogen encounters acidic pH, it becomes the active version of pepsin, which can then begin protein digestion. So it works together with the parietal cells. And then you have diffuse neuroendocrine system cells. Let's skip a slide. Oh, okay. Right, these are located at the bottom of the glands, closest to the blood vessels, gives hormone produced ready access to the blood, and you have G cells secrete the hormone gastrin, or we get gastric acid. Gastrin stimulates secretion from other cells known as like the parietal cells, along with the hormone histamine produced by different types of DNES cell, right? So stimulates the parietal cells. Remember, those parietal cells secrete hydrochloric acid, which helps the pepsinogen secreted by the chief cells to convert to pepsin, which helps with your protein digestion. And we're going to see that protein digestion is very important in the stomach. So these gastric glands are going to be very important for protein digestion. Right? So these are the different cells. Mucus at the top, protect the stomach, gastric pit, gastric glands, the parietal, the chief cells, and the DNES cells. And the mucosa neck cells secrete the mucus. What is the function of the stomach? Everybody kind of probably has an idea of what your stomach does, but we break it down into three primary functions: secretion. We just talked about some secretions. Propulsion, remember the movement, and digestion. Now we get into digestion, more into digestion. All right, so acid secretion from the stomach, the gastric glands we just talked about secrete multiple products. We talked about the hydrochloric acid, which is going to be released by the parietal cells, which will then activate the pepsinogen. A little bit more about that here. Hydrogen ions is created by the ATP consuming pump and the plasma membrane, drives hydrogen ion out of cell and into lumen of gland while taking potassium ion into cytosol. Remember, hydrogen plus chlorine. Chloride ion follows hydrogen ion via passive diffusion. Acid secretion occurs in this manner continuously throughout the day between meals. Right, so your stomach continually is secreting, between meals, secreting this hydrochloric acid. It's continually acidic. When you're eating, gastric acid secretion changes from a basal rate, this continual rate. Secretion can be divided into three different phases, right? Cephalic phase, gastric phase, and intestinal phase. Cephalic, we see some food, see some brown rice, some salmon, some grills. Actually, my wife just texted me. She said she wanted some fish tacos for dinner. So just the thought of those fish tacos had me salivating a little bit just now. Right. So hopefully I'll be able to make it in a way that it tastes good. And so when I set it in front of her this evening, she'll see it, she'll smell the salmon. I guess I should use salmon for the fish tacos. I could use another type of fish, but I don't know. I'm thinking salmon right now. So she'll see it, she'll smell it, eventually taste it. And like I says here, even the thought of it the stomach starts to prepare to receive the food by releasing hydrogen ions, which hydrogen ions are a part of hydrochloric acid. So that's your initial cephalic phase. So your body, your senses sort of picking up on food. All right, this stimulation causes a trigger from your vagus nerve, which is the main nerve of your parasympathetic, rest and digest, resulting in 4 physiological effects, which all increase your hydrogen ions, which are going to eventually increase your hydrochloric acid, basically. All right, so we have stimulation of hydrogen ion release, stimulation of gastrin. Remember, gastrin triggers your parietal, which triggers your hydrochloric acid, stimulation of histamine secretion, which also triggers hydrogen ion secretion, and inhibition of somatostatin secretion, which somatostatin is produced by a type of enteroendocrine cells located in the antrum of the stomach, which inhibits acid secretion. So we inhibit the somatostatin so we don't, because that would inhibit the acid secretion. So those are the 4 different steps that happen. And basically the idea there is to increase your hydrochloric acid. Then we go into the gastric phase. So cephalic phase, now the gastric phase begins when food enters the stomach. So we've increased this acid production, hydrochloric acid. Now food has traveled through your esophagus, the bolus enters the stomach. The gastric phase begins when food enters the stomach. Now we have 2 stimuli that trigger acid secretion during the gastric phase. The presence of the food, stomach walls begin to expand, distend, stimulates the neurons of your enteric nervous system, your ENS, and they notice this. There's food in here. Let's get to this. So both your vagus nerve and ENS neurons release acetylcholine, which also increases your acid secretion. Another stimulus is the presence of partially digested proteins in the gastric juice. So remember, the stomach is important for digesting a protein. So when they have the stimulus of detecting this proteins that are not digested, stimulates acid secretion, right? So this positive feedback loop progresses. Acid activates pepsin. Remember pepsin? We talked about that. Secreted by your chief cells. That pepsin gets converted. Pepsinogen gets converted to pepsin. Acid activates pepsin, which catalyzes protein digestion. Digested proteins stimulate more gastrin release, triggers more secretion of the acid, right? Once your pH reaches a certain level, which is 2.0, which is very acidic, negative feedback mechanism is initiated and triggers somatostatin release, which, remember we said that inhibits the secretion of the acid, so it decreases acid secretion. And that's what happens during the gastric phase. Right? So we have digestion, get too acidic, we stop releasing the acid. We talked about acid reflux. This term GERD, gastroesophageal reflux disease. The gastroesophageal sphincter, that's the basically the doorway between your esophagus and your stomach, normally is closed, except during swallowing when something's moving down, the bolus is moving down to your stomach. Now, but that mechanism can fail, just like any other part of your body can fail. And what would the problem would happen is you'll have acid from your stomach, hydrochloric acid, going up your esophagus. Now, when you regurgitate or when you throw up, if you've had that experience, you know what I'm talking about. But there are some people who have this happen often. And if that acid is continually going into your esophagus and into your pharynx and, other parts of your gastrointestinal tract that it's not supposed to go to, you can have some significant damage, pain, difficulty swallowing, vocal cord damage, respiratory problems, if that acid actually goes up your esophagus and then down your trachea. And so we want to neutralize that acid. And that's why we have antacids. Most common drugs contain alkaline substances that neutralize your acid in your stomach. All right, then we get into the intestinal phase, right? Cephalic, it's time to eat. Gastric, start producing acid, churning that food up. Intestinal phase, final phase of the gastric acid secretion, responsible for only about 10% of remaining acid secretion, right? Triggered by the presence of partially digested proteins, once again, similar to the previous phase, proteins and fluid entering the duodenum. Now, partially digested proteins entering the duodenum, the first part of the intestines, which we're going to talk about, triggers enteroendocrine cells to release intestinal gastrin, right? Gastrin from your intestines. Same effect, stimulates hydrogen ion secretion from parietal cells. That's how we get our hydrochloric acid. As chyme enters your duodenum, declining pH and presence of lipids trigger enterogastric reflex, which decreases your vagal activity and acid secretion. So we don't want this area to be as acidic as it is in your stomach. So as the pH declines and also the presence of lipids, remember fat, lipids, enter your intestines, we have this enterogastric reflex, which causes the decrease in this acid secretion. The low pH in the duodenum also triggers the production of hormones by cells of duodenal mucosa, which we're going to talk about the structure of the duodenum and the rest of the intestines, including secretin and gastric inhibitory peptide, which reduce acid secretion. So we only have in the intestinal phase, like you said, it's triggered by partially digested proteins. There is some acid release, but when the pH drops, we're quick to shut it off once it gets into the intestines. We don't really want the same effect as we had in the stomach of, just maintaining an acidic environment. All right, so this is the intestines. The stomach's motility enables it to perform three actions. You receive the food at the esophagus, so passing through the esophageal sphincter, turns the incoming bolus. We go from bolus to chyme, and we also control the way in which chyme empties into the small intestines, right? So we have a control mechanism there. In its resting rate, stomach has a volume of only 50 milliliters, but it can expand to 1,500 milliliters when filled with food and liquid. So your stomach has we said is distensible, has a huge ability to expand. All right, we have the ability for the stomach to fill up, we talked about. And it talks about receptive relaxation here. But important, this last point is that as increases in gastric pressure can trigger vomiting, expelling of stomach contents through mouth, you have to be, this is all mediated by your medulla and your vagus nerve, right? So the stomach can only get so big, and then obviously if you kept trying to push stuff into it, you would eventually your body would eventually regurgitate it back up. And so it allows your body, your stomach to relax, allows it to fill up, but it's controlled by the medulla and the vagus nerve so that it doesn't get too full so you don't throw up. We have this churning function of the stomach. We talked about this term peristalsis, which is controlled by smooth muscle. And it's initiated by a group of specialized cells known as the gastric pacemaker. And we have this ability to, and this is another thing that is kind of cool to maybe watch a video on to see how, the three layers of the three smooth muscle layers, how the muscles of the stomach can churn and creates this, we go from this bolus to this chyme. And this peristalsis prepares the bolus to the pylorus, to the end of the stomach, eventually to the pyloric sphincter and the duodenum, the first part of the intestines. Whereas parts of the chyme are pushed backward, where it continues to churn. This process just repeats. So it gets this, so the stomach basically fills up, starts churning, and pushes little bits at a time down your intestines. There's a couple of hormones involved. Serotonin stimulates gastric motility, and then you also have intestinal gastrin, which also stimulates gastric motility. Then we have the emptying function, final function, control movement of the chyme into the duodenum. Different materials pass through the pyloric sphincter at a different rate. So liquids go through pretty quickly. They don't need to be churned up. They're already pretty much broken down, able to be absorbed. Whereas something that is like meat, for instance, would take longer in your stomach to be converted into the liquid chyme and then be passed on through the rest of your small intestines. Right? It has to be in like a near liquid state before it gets into your intestines. I thought it was it's a good slide to pay attention to. Control of your gastric emptying is critical because duodenum must mix incoming chyme thoroughly before it moves to the rest of the small intestines. So the chyme is acidic. The duodenum mixes it with bicarbonate. Remember, we talked about this bicarbonate buffer system before with the blood, and this bicarbonate is also used to sort of, remember, this chyme that is made by the stomach is acidic, and so we need to sort of neutralize it, and so we have bicarbonate to do just that, so that it doesn't damage the rest of your intestinal tract. And also must be diluted with water. That helps also make it fluidic as well and easier for it to pass through your body. So the chyme is generally very constant, must be diluted with water from pancreatic juice to prevent chyme from drawing water into intestinal lumen by osmosis. We're going to talk about that process because that's going to be as we get into the absorption process. Right. So, and the duodenum, as we talked about, receives portions of the chyme at a time. Ooh, that rhymed. And, because it only can process certain small amounts at a time. So portions get pushed into the duodenum, and then the rest goes through the churning and the peristalsis, and then a little bit more gets pushed in, and it goes back and forth. All right, and that's just showing you how that happens. Now we go into the small intestine. So we've gotten out of the stomach, we've gone from bolus to chyme, and now we go into the small intestines. We've gone from super acidic in the stomach. We try to reduce the acidity once we get into the intestines, and now we will get into further digesting and into absorption. All right, so intestines, longest portion of your alimentary canal. They're almost 20 feet long, your intestines, right? So think of a long winding tube, 20 feet long, longer than you are tall. Isn't that kind of cool? But it's all folded over. 4 main processes occur here. You have secretion, similar to the stomach, digestion, breaking it down, propulsion, moving it through, moving the chyme through. But then we also have absorption, right, which is going to be very important, going from digestive system to cardiovascular, to our bloodstream, right? So the cells of the small intestines are the enterocytes. You'll see this term, which produce multiple digestive enzymes, hormones, and mucus. Mucus normally is for some type of protection, right? Enzymes break down the food, hormones to allow some certain processes to continue to happen. So enzymes, along with those released by the pancreas, we're going to see that the pancreas starts to get involved here. We're going to see involvement in the pancreas. We're going to also see involvement from the liver coming up really soon. Enzymes, along with those released by the pancreas, are responsible for the bulk of the chemical digestion. Remember, digestion is chemical and mechanical, but those released by the pancreas are responsible for the bulk of the chemical digestion. So no further nutrient digestion takes place by human enzymes once food leaves the small intestines. That's an important point to recognize. No further nutrient digestion takes place by human enzymes. And the key term there is human. We're going to talk about something else that happens that lives in your gut, in your large intestines, but we'll talk about that when we get there. After nutrients are digested chemically, they are absorbed across your enterocytes into either the blood, lymph. Remember we talked about absorption into the lymph a little bit, along with water, vitamins, and other substances. All right, so divisions of your small intestines, we break it up into 3 different major divisions. And they have different histological differences, remember histology, study of tissues, different tissue differences, right? That's how it's not necessarily just the location, but it's also differences in tissue, right? But location is the easiest way that we recognize it, because so the beginning, the center, and the end. All right, so duodenum, that's the first initial segment, begins at the pylorus. So right after that sphincter, you have the shortest of the 3 divisions, the duodenum, only about 10 inches long. Remember, at 20 feet, this is only 10 inches long, arches into a C shape, curves around the pancreas, where only proximal portion of duodenum is within the peritoneum. Remainder sits posterior, houses major duodenal papilla, where secretions from the gallbladder and the pancreas enter your small intestines and play a crucial role in digestion, right? So this is where secretions from your gallbladder and pancreas enter the small intestines. And you have this duodenal submucosa contains specialized glands called the duodenal glands, which produce this alkaline mucus to protect the duodenum from this acidic chyme. Remember, this chyme is still acidic because it's just come from the stomach. Then we have the jejunum, right? Begins at the duodenojejunal flexure, right? And this part is 7.5 feet long. So we go from 10 inches to 7.5 feet. This is the most active site for chemical digestion and absorption. So we have digestion and absorption here. And then we have the last segment, duodenum, jejunum, and ileum. Final segment, 10 feet, 10.8 feet in length, terminates at your cecum, which is the large intestines. You have the ileocecal valve. Notice another valve, another sphincter that controls an opening. So a lot of controls here from the at the opening of the esophagus, from the pharynx, the esophagus, from the esophagus, esophageal, from the esophagus to the stomach, from the stomach to the duodenum to the intestines, and then from the small intestines to the large intestines, the from the ileum to the cecum, ileocecal valve. Controls movement of materials from the ileum into the cecum. All right, prevents materials in large intestines from flowing backwards into the ileum. Remember, we don't want it to go backwards. Notice that theme, which is also critical for preventing bacteria. There's bacteria that live in your large intestines we're going to talk about from going back into your small intestines. Right, and that is the structure of your, it's a nice drawing of your small intestines, which leads into your large intestines. Okay. Now let's get into the function of your small intestines. So internal surface of small intestines contains 3 progressively smaller types of folds. Remember these enterocytes, remember those. Most absorption takes place in this region, right? So intestines is where we have absorption. Folds, you have these folds in the intestine surface, increases the surface area of the intestines, which makes it more likely for nutrients to come in contact with that surface, eventually get absorbed into the intestines, through the intestines into your bloodstream, into your lymph, etc. You have different types of folds. Largest types of folds are called circular folds, visible to the naked eye. Involve both mucosa and submucosa, and the two innermost, the epithelial layer and the connective tissue layer. The small intestine increases surface area, makes it slow down the transit of something through. So if something is bumpy, the chyme is going to go slower as it goes over these folds, give the nutrients more time to digest, and through the enterocytes, which are those cells of the small intestines. There's also smaller folds within these bigger folds, which are not visible. And mucosal folds is the projections called villi or villi, depending on how you want to pronounce it. Contain layers of enterocytes surrounded by blood capillaries and lymphatic vessels. Between villi and mucosa indents to form intestinal crypts, which can house glands, which have both enteroendocrine glands and also mucus secreting glands. This is where we get the mucus, which helps protect. Right, so this is your sort of your circular folds. And then within those circular folds, you have the villi, more folds, increase and more surface area. And this is the enterocytes, which then you're going to lead into the blood vessels and lymphatic vessels, right? The red arterial, blue venous, green is your lymphatic. Smallest folds, microvilli, are in plasma membrane of enterocytes. Enterocytes means 3,000 microvilli, gives cell appearance of bristle, right? We're just further folding, increasing that surface area. So we want as much surface area as possible. Digestive enzymes produced and secreted by enterocytes catalyze reactions that break down disaccharides, carbohydrates, and peptides, right? Disaccharides are just, remember, monosaccharide, disaccharide, polysaccharide, all right? So we go from big polysaccharides to smaller polysaccharides by the saliva, and then we further can break down disaccharides in our intestines and also peptides. Right. So that's the structure there. All right, motility. All right, so we have these folds that create a lot of surface area. We are able to further digest, break food down. We can absorb. We're going to go into more detail on absorption. It's all happening in your intestines. We also have motility. Remember, things has to keep moving and moving in a specific direction, you know, from the mouth, esophagus, stomach, small intestines, large intestines, to the anus, right? And absorption happens along that path, but we don't want things to go backwards. Remember that. All right, so during fasting, small intestine exhibits slow rhythmic contractions along its length and pattern called migrating motor complex. So when you're not eating, contractions can clear any remaining material. It's, you know, slowly moving. Any leftover food can be pushed out of your body. It requires about 2 hours to push digesting food from the duodenum, start of your small intestines, to your ileocecal valve, your large intestines. And that's, you've probably heard, when you eat something, they used to say you should wait about 2 hours before you exercise or jump in the pool and go swimming, things like that, because if you're in rest and digest, parasympathetic nervous system is at play, you are digesting food. It takes time for that food to go through your body system before it digests and gets absorbed. And so blood is flowing to your stomach and your flowing, well, to your stomach is working, your intestines are picking up or digesting as well as picking up nutrients. And so we want the blood going there and not towards your muscles, right? So this is one of the reasons why they say once you eat, to rest, basically, and allow your food time to basically, we say digest, but what we mean is digest and absorb and move through our system, basically. This is controlled by the ENS, enteric nerve, you're part of your autonomic nervous system, and motilin, a hormone produced by cells in your duodenum mucosa. So what happens? We have peristalsis, very similar to what happens in your stomach. Alternating contractions, longitudinal and circular layers of the muscle, which propel the chyme towards the ileum, and it ultimately through the ileocecal valve. So propels the food that you've eaten, that is turned from the bolus to the chyme in the stomach, propel it through the intestines. And we also have segmentation, contractions of only the circular layer of the smooth muscle, which squeeze. Remember, like you're pushing something through a tube, squeezing motion. Primary functions of segmentation are mechanical digestion, so further breaking it down mechanically, and mixing the chyme and intestinal and pancreatic enzymes, as well as bile, which we're going to talk about from your gallbladder. So mixing it up and further digesting it. All right, so peristalsis and segmentation. Peristalsis, and then we have just the circular smooth muscle, sort of mixing it up to further digest. And this is the motility, the movement of the chyme through the small intestines. And this is a chart just showing you the hormones and paracrines involved. You can read through this on your own. Appendicitis. Some of you all may not realize where your appendix is, but it is very much involved, not very much involved. It is very much a part of your digestive system. All right, small size, it's blind-ended. So that means things can only go in one end. And occasionally it can become blocked by fecal matter, which can cause problems because bacteria which is in our feces, those bacteria which live in our large intestines, which we don't want to go back into our small intestines or stomach, can multiply in the appendix and cause infection, which will result in appendicitis. This usually shows itself by large pain, um, tender, nausea, vomit. And what happens is you don't want your appendix to rupture. That is actually life-threatening. So what they do is, if they recognize in time that you have appendicitis, they remove your appendix. Because if it were to rupture, the bacteria from your fecal matter would spill into your peritoneal cavity, which could cause all kinds of inflammation and issues and problems.